My name is Yui Takeshita. I'm a scientist at the Monterey Bay Aquarium Research Institute, and my portion of this workshop is to tell you about the biogeochemical sensors on the floats and also the methods and protocols that we use to adjust BGC data as well as validating those adjustments. And before I start, I want to acknowledge the sponsors, NSF, NOAA, NASA, and OCB and US CLIVAR for supporting this workshop. Okay. Okay, so as mentioned before, currently there are 5 parameters, biogeochemical parameters that are equipped on GO-BGC floats: oxygen, nitrate, pH, and then also chlorophyll A fluorescence and backscatter, and those are measured by a single sensor, so that's why they're in the same color. And irradiance is soon to come. We have our first test float with irradiance sensors on them getting deployed next month, so hopefully this will be a addition onto the GO-BGC float array in the near future. And so I kind of want to, before I really start the talk, I want to kind of emphasize the point that this is, there's a network of sensors on each profiling float, and they really, complement each other and provide more information than if, like in any of these if the float didn't contain all the sensors simultaneously. And this idea, there's a name for this, and this is it's called sensor fusion. Basically, it's the idea that if you combine data from multiple sensors and they work synergistically, then you can either reduce the uncertainty or gain more information than any one sensor or subset of the sensors can provide themselves. And then the BGC flows are such a beautiful example of this, because you'll see how some of the sensors, for example, most of the BGC sensors need the core temperature, salinity, and pressure data to make accurate measurements. And not only that, some of the adjustments that are required rely on accurate measurements of other sensors. So that's this, you get a sum that is bigger than the individual components. Okay, so in this talk, I'm going to briefly talk about the technologies, the sensing principles behind the 5 sensors. I'm not going to talk about irradiance because those are not deployed on our floats yet. So there are recorded talks online for oxygen, nitrate, pH. Unfortunately, I didn't have time to do the bio-optical sensors, but hopefully you will get one of those videos in the future. But this can be a brief summary, and then I'm going to talk about how we adjust BGC float data before they are disseminated to the community. Okay. So as Steve mentioned, there are basically 2 types of floats that we currently deploy: Apex and the Navis, and then soon to be the BGC Solo 2s. And so they both will carry the same sensor suite, but they have slightly different sensors on the 2 floats. And for most of them, they're interchangeable and the performances are very comparable, except for the oxygen sensors currently. And then so the Apexes use on their optodes, shown on the left, and then Navis floats will currently use SB63, which is integrated into the pumped flow stream of the CTD. So the benefit of this is that it has a much faster response time than the on there, but it's not capable of doing air calibrations, which I'll go into in a second. So that's the downside of the Seabird 63, but there is currently a sensor in development, in the final stages of development of Seabird, which can both be pumped and do air calibrations. And we have deployed 4 of those sensors so far this year, and the initial results are very encouraging. So hopefully that will be, they'll kind of become the standard sensor for most of our floats moving forward. Nitrate sensors for Apex floats, they are equipped with ISIS, which is integrated into the float end cap, as you can see on the left. And then for the Navis and the BGC Solos, they used Suna, which is the commercially available version of this, and it's bracketed to the side of the float. pH is basically identical between the 2 floats. Very, very small mechanical differences, but they can just be considered the same or synonymous. And finally, bio-optical sensors. The Apexes use FLBBs that are bracketed to the bottom part of the float, whereas the Navis floats use MCOMs, which is integrated into the at the top of the float. But the performance of these 2 sensors are very comparable as well. Okay, so now we're going to walk through the sensing principles for the various sensors. So we'll start with oxygen. So the oxygen sensor, or the optodes, use a luminescence quenching to measure oxygen. So the idea is that So if you imagine this box as the membrane of the sensor, so there are these compounds called luminophores, and so when you hit them in some blue light, they get excited, and then they emit red light. They return back to its original rest state. And over time, as it, but this decay takes a while, especially if there's no oxygen. So this is the decay, the E-folding decay time of the sensor. But if there's oxygen present, shown in the blue here, then the luminophore can actually transfer some of its energy to oxygen molecules directly without luminescing. Therefore, it reduces the decay time. And then we measure this difference using a phase shift. And so that's the analog signal, and then we use the Stern-Balmer equation to calculate oxygen concentrations. So, this, again, all the details for this is in the pre-recorded talks. Nitrate sensors measure nitrate using UV absorption. So nitrate absorbs in the deep UV. This is the absorption spectra from 210 to 240 nanometers. And but the complication is that other compounds in seawater absorb in the same range as well, so namely bromide, and also dissolved organic carbon, which we parameterize as a linear fit. And so when you actually measure the absorbance of seawater in this range, it's a sum of these three components. And so the trick really here is how to deconvolute these three spectra, so you can subtract out what the nitrate concentration is. And the approach that we take is actually to calculate out what the bromide spectra should be based on temperature, salinity, and pressure data. And then so you can subtract that out from the measured absorbance. And then now you only have to fit two parameters, which is much more accurate. pH is measured using the Honeywell ISFET, and it's a potentiometric sensor. So basically what that means, so this is a picture of the deep sea Durafet. And so potentiometric sensor is you measure a voltage between two electrodes. So the first electrode is the ISFET, or ion sensitive field effect transistor, manufactured at Honeywell. And this electrode is sensitive to pH. And then the other electrode is a reference electrode, which is ion selective electrode for chloride, shown in the red arrow here. And that basically, it responds to chloride ions or salinity. And so you measure the voltage between these two electrodes, and then you calculate pH using the Nernst equation. And this, and each sensor is individually calibrated for temperature and pressure, so that we can, calibrate for over temperature and pressure ranges. Chlorophyll A fluorescence, this is a very there's a long history, and there's a very established technique. Basically, chlorophyll fluoresces if you hit it with blue light, and then fluoresces with red light. And so this is thought, then the fluorescence can be related to the concentration of chlorophyll A, which can then be a proxy for how much phytoplankton there is in the water. And then in practice, you modulate the excitation light source to kind of filter out some of the ambient noise in the light. And currently, these sensors use an excitation wavelength of 470 nanometers and measure the emission as 695 nanometers. So actually, the sensing technology is very, very robust for fluorometers, because it has a very long history. And it's a very mature sensor, but the challenge for this sensor really is then how you convert fluorescence, which is what the sensor measures, to properties that you care about, which are chlorophyll concentration, or and ultimately to biomass. And so I will kind of walk through that towards the end of the talk. And the final sensor is optical backscatter, and so this is a proxy for particle concentration, or particular organic carbon in particular. So you can imagine a light source on the right here and a detector on the left, and then so the light source emits light at a known angle, and then the particles will scatter light, or just reflect light in a bunch of different directions. And backscatter refers to the part of the light that is scattered between 90 degrees and 180 degrees from the incident light angle. And so the detector measures a part of this backscatter and integrates it to the whole scattering volume. And then from that measurement, we can calculate something called BVP, which is a backscattering coefficient. So that is a direct sensor output. And then we use relationships that we observed in the ocean to go from backscattering, the BVP, to POC, or particulate organic carbon concentrations. Okay, so that is a very, a crash course on how all of these sensors work. And so now we move on to the data adjustment and validation part of these sensors. And so data adjustment is a really, really important part of operating biogeochemical sensors effectively and properly, because most of these BGC sensors tend to they can suffer from calibration offsets at the time they're deployed, and also drift over time. So they're not quite the plug and play sensors that we expect for the CTBs. And so in order to produce science quality data that can be used for operational or research applications, adjusting the data is really, really critical. And not only the adjustments, but also validating those adjustments as well. And then so all of the data that we serve on the Argo data centers are adjusted in real time, and so that the community and the users can use it, knowing that the data they're adjusted accurately. And then so I'm not going to go into where and how to access these data, but Tanya will talk about that in a few minutes in the data management part of the talk. Okay, so I want to mention, so SOCOM has been brought up, but the SOCOM project has deployed over 200 BGC profiling floats in the Southern Ocean over the past 6 years. And so a lot of the methods that were developed that I'm going to explain here on adjusting the sensor data comes from the knowledge and the data obtained for SOCOM, and it really wouldn't have been possible without this program. And so SOCOM made a tremendous effort to collect discrete samples each time the float was deployed. And then so that gives us an unprecedented data set where we can actually compare float measurements to discrete samples to validate or test and validate these adjustment protocols. And I want to emphasize that the discrete samples were not used to calibrate or adjust the data, and they represent an independent validation for the data adjustment. And I'm going to show you a bunch of data from this data set. Okay, so now we have to walk through each individual sensor and parameter because they are adjusted in different ways. And before we do that, so data adjustment can basically take place in two ways. One is by applying an offset, and second is applying a gain. And then an offset is basically when we can correct the data, but just adding an offset value. So it's an additive correction. So the corrected value is the raw sensor output plus an offset. And gain is when you multiply the raw sensor output by some gain factor to get the corrected value. And some sensors only require an offset, and some sensors only require a gain, and some sensors require both. So to keep track of that, I'm going to update this table as we walk through all of the different sensors. So starting with oxygen. So oxygen, we correct oxygen data, but just applying a gain correction. And to do this, we take advantage of the fact that the flow comes up to the surface to telemeter data every profile. So during the telemeter phase, the octode here can make measurements in air, and it turns out the octode has the same response in air and in seawater because the sensor responds to partial pressure of oxygen. So what that provides is if we can calculate or know what the partial pressure of oxygen is in the atmosphere, then that will serve as a really great reference measurement for this sensor, and then we can use that reference measurement to adjust the data. And it turns out, fortunately, that the partial pressure of oxygen can be calculated pretty straightforwardly because the molar content, or the percentage of oxygen molecules in air, is constant at around 21%. So if you know the total atmospheric pressure, which we can estimate using model or reanalysis data such as NCEP, we can calculate what the PO2, or the partial pressure of air, is in the atmosphere, and then compare that to the sensor reading. And the gain is simply just the ratio of this calculated partial pressure of oxygen divided by the sensor reading in the air. So, very straightforward. And we can apply this gain in either PO2 or the constant oxygen concentration domain to adjust the data. And another benefit of these air calibrations is that you get a measurement every profile, so we can assess for sensor drift over the life of the float. And oxygen sensors tend to be very stable over time, but they do exhibit some drift over multiple years. And so since the signal is really small, we currently only correct for drift after 2 years of only if 2 years of data is available, and typically, and only if the drift is significant, and typically these sensors drift by less than 0.5% per year. But if the float lives for 5-6 years, then that can add up to be a pretty significant source of bias. So we will correct for that. So this is the plot that compares the bottle oxygen versus float oxygen. Over 2,000 points, measurements were made, and you can see that they follow a very nice one-to-one relationship. And then the median residual between this data set is 0.35 plus or minus 7 micromole per kilogram. And if you assume that about half of this variability is due to the spatial temporal discrepancy between the float and the bottle data, we can assume an accuracy of about plus or minus 3 micromole per kilogram. I'm already out of time. Oops. Okay, so nitrate and pH. So we adjust both of these sensors by applying an offset. So basically, for this, we use the deep measurements at 1,500 meters because the deep ocean is fairly stable and also predictable. And so if you have reference measurements in the deep ocean, then we can compare that against the sensor measurements and correct for drift. And then so how we do that is use empirical algorithms with the inputs of temperature, salinity, oxygen, location, depth, and date. And the output is estimated pH or nitrate and with an accuracy of better than 0.007 or half a micromole per kilogram for pH and nitrate. And then we can calculate, we can compare this estimated parameter to the measured parameters and then calculate this anomaly. And this anomaly is essentially the sensor drift at depth because we assume that the estimated values are accurate. And then so how we implement this, we don't make a correction every profile, but we only make corrections once the anomalies go outside of some confidence interval. So if the anomaly is inside of this confidence interval, we don't make any adjustments to the data. But once this anomaly starts drifting outside of the confidence interval, which is plus or minus 1 micromole for nitrate and plus or minus 0.01 for pH, I believe, then we correct the data using a linear regression. So we only correct the parts of the data that start drifting. And it's not uncommon to see jumps in the data, particularly for nitrate sensors. And to accommodate for this, we do a piecewise regression so that we correct different parts of the data separately and then correct it all to so the anomaly equals 0. Okay, and the validation, nitrate on the left, pH on the bottom, but basically with using the same line of argument, we think the accuracy of the nitrate sensor is about half a micromole per kilogram and pH to be about 0.007. Okay, and then for using pH, we can calculate derived parameters for the carbonate system as well, and we estimate the alkalinity using an empirical algorithm with the same inputs, and the algorithm can estimate pH to about 6 micromole per kilogram, and you combine that with the measured pH, and then you can derive parameters such as DIC, saturation state, and PCO2. But just one thing that I want to mention is that we use and measure pH directly for DIC and saturation state, but for when we calculate PCO2, we apply an additional correction. And so this comes from the fact that pH and PCO2 aren't exactly internally consistent, or so there's some lack of knowledge in the CO2 system. And yeah, so we can see the talks for more details on that. Okay, I'm going to skip this slide. Okay, so going on to chlorophyll. Chlorophyll requires both a gain and an offset correction. And so chlorophyll, the sensor measures fluorescence, and you need to go to concentration. And so the offset really only comes from when we measure the fluorescence. So the fluorescence is the counts from the sensor minus the dark counts, which is essentially what the reading is when there's no chlorophyll present. And so this represents the offset adjustment that's applied to the sensor, and then we can calculate what this offset is based on deep measurements where there's no chlorophyll available. And then the gain correction comes from when we want to go from the chlorophyll A fluorescence to concentration of chlorophyll because we need to multiply this fluorescence against the scale factor. So that's from the gain, and we try to establish a scale factor by comparing sensor measurements to concentration of chlorophyll by making discrete measurements using HPLC. But the problem is that this gain factor is very, very variable. And then so a paper by Rossler in 2017 measured this gain factor globally. So this goes from the Arabian Sea to the Southern Ocean. So as you can see, this scale factor can change from 1 to over 6 depending on where you are. And then so they noticed that the global average is 2, and then so recommended that this gain factor 2 is used for floats from a global perspective. But you can see there'd be a large uncertainties if you're depending on if you're down here or up here. You can your estimated chlorophyll concentration could be off by a factor of 2 or so, or 3. And so this result was validated using the SOCOM data as well. So this is the gain based on SOCOM data, and then the mean is about 5. And mean is about 5, but it can range from about 1 to 10. So depending on depth, season, and location. So this gain factor is very, it's large variability. And so trying to determine what gain factor to apply is a current active source of research. A final question we have to make for chlorophyll fluorescence is non-photochemical quenching. So during the daytime, the efficiency of fluorescence goes down, and then so therefore, so even if there's the same concentration of chlorophyll shown on the x-axis here, in the nighttime, the fluorescence will artificially be biased low because of this NPQ phenomenon. And then so in order to correct for this, we use the approach in Xing et al. In 2012, where we basically find the mixed layer depth and then find the chlorophyll max above the mixed layer depth, and then assume that it's well mixed above that, and then so we just extrapolate straight up to the top of the water column. We do this during the day, and so this is an imperfect correction. So depending on where this chlorophyll max is in relation to the mixed layer depth, can lead to biases in this correction. So that's another thing to keep in mind when using the adjusted chlorophyll concentration data. Okay. Finally, BBP or backscattering. So backscattering, we correct the sensor data by applying an offset, and this accounts for the 0. And I went over that for chlorophyll, so that's the same idea. And then the adjustment or the derived parameter that we calculate from BBP is POC, or the particulate organic carbon, which is a proxy for biomass. And so we have collected POC samples from Niskin from the time of deployment shown on the x-axis, and the y-axis is the measurement from the flows. And you can see that there's a pretty good relationship between these two. And the relationship that we established in SOCOM is showing this light blue, but all the other lines are different models from around the world. So there seems to be a pretty good and coherent relationship between BBP and POC around the world. And then so kind of averaging all of this, we also report POC that's derived from BBP using these relationships. And we think the uncertainty for this is about plus or minus 20% or 35 millimoles per meter cubed. Okay, I think this is my last slide. Okay, so this is the last slide. I think I'm out of time, so I just want to say thank you very much. And then I hope you learned some of what is behind the data adjustments, and hopefully we will turn this dashed line of radiance into a full circle in the near future. And thank you very much.